Good day, future doctors. Today, we will be discussing the introduction to a group of parasitic helminths we refer to as trematodes. Medically important parasites, or those parasites that can cause diseases in humans, are grouped into protozoans and parasitic worms. These parasitic worms are further grouped into nematodes. We call them as roundworms. Then we have the trematodes and cestodes. Cestodes and trematodes are classified under the phylum Platyhelminths. Platyhelminths mean flat worms. Take a look at the pictures of a cestode and a trematode. Both of these worms are flatworms. What sets them different from each other is that the bodies of cestodes are segmented, just like what is shown in the picture. And since the bodies of cestodes look like that of a measuring tape, these worms are referred to as tapeworms. Trematodes, on the other hand, are flatworms, meaning they are dorso-ventrally flattened, similar to the cestodes, but their bodies are unsegmented. Trematodes are also known as flukes. Why? Because these worms basically look like that of a fluke fish. The fluke fish is dorso-ventrally flattened, and it is also leaf-like in shape, just like most of the trematodes. Trematodes are also known as flukes, and they are classified under the phylum Platyhelminths because just like the cestodes, they are dorso-ventrally flattened. To differentiate them from cestodes, you must look for the segmentation of the bodies, which are not seen in flukes or trematodes. Their bodies are leaf-like or oval or lancet shape. All members of the trematode group are dorso-ventrally flattened, with one exemption. And that exemption is the Schistosoma species. So therefore, we can conclude that all members of the trematodes, especially those that are medically important, are dorso-ventrally flattened except for the Schistosoma. Take a look at the picture of a typical trematode. It has a leaf-like shaped body, and its body is dorso-ventrally flattened. Take a look at the picture of Schistosoma. It resembles more of the nematodes or roundworms. But there's a reason why these Schistosoma species are classified under the trematode group, and we will discuss that later on. At this point, you have to memorize that all parasitic trematodes to humans are dorso-ventrally flattened except for Schistosoma. Another difference between a typical trematode and the Schistosoma species is that the eggs or ova of typical trematodes are operculated, and you don't see the same structure in the ova of Schistosoma. What you have on the Schistosoma eggs are the spines attached to the shell. What have we learned so far? That all parasitic trematodes to humans are dorso-ventrally flattened except for Schistosoma. All trematodes have operculated ova except for Schistosoma. Another exemption is that all parasitic trematodes are hermaphrodites except for Schistosoma. Take a look at the picture of a typical trematode. Take a look at the structures labeled as ovary and testis. This worm is hermaphrodite because it contains both the male and the female reproductive organs. While Schistosoma species have separate sexes, they are described to be dioecious, meaning you have a male and a female worm. All parasitic trematodes are hermaphrodites except for Schistosoma because Schistosoma species are dioecious, meaning they have male and female worms. The larger worm in the picture is actually the male, while the longer and thinner worm is the female. And the female is usually enclosed in a canal situated in the midline of the male worm. Let's try to study the life cycle of a typical parasitic trematode. The human host harbor the adult or the sexual stage of the parasite. Therefore, humans act as definitive hosts. These adult worms will lay eggs and they will be discharged out from the human body. These eggs will later produce a ciliated stage and we will call it as the meracidium. This meracidium will infect snails, and inside the body of the snails, the meracidium will become the sporocyst. The sporocyst will develop into the rhidia, and the rhidia will produce the tailed stage, which is the cercaria. And what will come out of the body of the infected snail is the tailed stage cercaria. The cercaria will infect another host. And in the case of this parasitic trematode, it's an aquatic plant. And since in the snail and in the aquatic plants, you only have there the larval stages of this parasite, therefore, the snail and the aquatic plants are acting as intermediate hosts. So in the life cycle of a typical parasitic trematode, you have there two intermediate hosts. The first is the snail, and the second, in the case of this trematode, is an aquatic plant. What will happen now to the cercaria once it will infect the second intermediate host? It will actually form a cyst. It will insist itself into the body of the second intermediate host. And it will transform into another stage. And we call that as metacercaria. Eating the raw second intermediate host and harboring the metacercaria will predispose the human host to being infected by the trematode. So the infective stage of a typical parasitic trematode to humans is the metacercaria. Let's study the life cycle of the Schistosoma species. The human hosts harbor the adult stages. So therefore, the human hosts are acting as the definitive hosts. And what will be discharged out from the body of the affected human host is the egg or ovum. The egg will later release the ciliated stage, which we will call as the meracidium. And this is now the reason why, even if Schistosoma species resemble more of the nematodes, they are still classified under the trematode group because they have the same stages like that of the trematodes. Now, what do you think will happen to this meracidium? This meracidium will infect snails. Inside the body of the snail, you have there the development of the sporocyst. This sporocyst will release the tailed stage, which is the cercaria. But take a look at the life cycle. There is no second intermediate host. So this Schistosoma species will not produce the metacercaria stage. Instead, this cercaria will penetrate the skin of the susceptible human host. So therefore, in Schistosoma life cycle, you only have one intermediate host. You don't have the metacercarial stage, and the infective stage to humans is the tailed stage cercaria. How do we acquire infection of the typical trematode? By eating raw second intermediate host. How do we acquire the infection caused by Schistosoma? Skin penetration by the cercarial stage. Therefore, these are the additional exemptions. All parasitic trematodes have second intermediate hosts except Schistosoma. All have metacercarial stages except for Schistosoma. All are acquired through eating raw second intermediate hosts except for Schistosoma, because schistosomiasis is acquired through skin penetration by the cercaria. What is common to all is that they all have the same first intermediate host, and that is the snail. But mind you, they affect different species of snails. With regards to the characteristics of their eggs, I want you to take note that some will produce embryonated ova, meaning once their ova will come in contact with water, it will readily release the miracidium, while some will produce unembryonated ova, meaning the ova have to stay first in water for quite some time before it will be able to form the miracidia stages. Now, what trematodes are producing unembryonated ovum? You have to memorize the acronym PEFF. All trematodes lay embryonated eggs or ova except for PEFF. P. Paragonimus. E. Echinostoma. F. Fasciola. F. Fasciolopsis. So, trematode worms that are not included in the list would produce embryonated ova. Take note, these 4 trematodes have eggs that cannot be morphologically distinguished from each other. So take note of that. That will be very, very useful in the succeeding discussions. This is the life cycle of Fasciola, and Fasciola is one of those trematode worms that will lay unembryonated ova. That's why, according to the number 1 part of the life cycle, what are discharged from the human body are the unembryonated ova. And those unembryonated ova would have to stay first in the water before they will be able to produce the miracidial stage. This is the life cycle of Clonorchis sinensis, and this life cycle is almost similar to the life cycle of Opistorchis. These 2 parasites are able to produce embryonated ova. And there's a reason why that these parasites should produce embryonated ova. The snails that they are supposed to infect are terrestrial snails, and these snails would prefer to stay more on the land rather than in the water. So just try to imagine if they will produce unembryonated ova, the unembryonated ova might just hatch anytime and release the miracidia. If the snails are not on the water during the time the miracidia are released, then these miracidia will just die without infecting the susceptible snail host. So to make sure that the life cycle will not be disrupted, these parasites will lay embryonated ova. So meaning the miracidial stages are already inside. And they will wait for this embryonated ova to be washed ashore. And what will happen? If the terrestrial snails will go to water to feed or to take in water, they might accidentally ingest the embryonated ova of Clonorchis and Opistorchis, and that will ensure that the miracidium will not die. It will really reach its susceptible snail host. So if you will be asked in the exam, what is the infective stage of, let's say, Fasciola to its snail host, the answer is the miracidium. But in the case of Clonorchis and Opistorchis, the infective stage to the snail host is not the miracidium, it's the embryonated ovum. Once the embryonated ovum is ingested by the terrestrial snail, it will open up its operculum inside and release the miracidium, and the life cycle will continue. We will now discuss the different structures of trematodes which can aid us in identifying them in the laboratory. Let's start with the structures that will enable these organisms to attach themselves to the different tissues or organs of the susceptible human host. We have the oral sucker and you have the ventral sucker. Before we'll proceed to the next slide, I want you to remember the location of these two suckers, oral sucker and the ventral sucker. As for the structures that they use for attachment, they are provided with two types of suckers or acetabula. You have the oral and the ventral. These parasites are also provided with spines on their integument, which will also aid in attachment. This is a picture of a fluke or trematode. This is the oral sucker. This is the ventral sucker. Take a look at the pictures of this trematode. On their integument or body surfaces, you have there the presence of spines, which they can also use for attachment aside from their oral and ventral acetabula or suckers. This is the oral sucker, and this is the integument of the worm. Let's take a look at the integument at a higher magnification, and this is how it will look like. And then, this red arrow is pointing to one of the integumentary spines. You have learned a while ago that for attachment, these worms will use their spines and two types of suckers. You have the oral and the ventral suckers. But there's that one particular trematode that is provided with a third sucker. And we call that as the genital sucker. And that particular worm is the heterophyes worm. And the main purpose of this genital sucker is for the release of eggs. This is the picture of a heterophyes worm. Number 1 is oral sucker. Number 2 is ventral sucker, while this sucker pointed by the red arrow is the genital sucker. Take note, this is also known as gonotile. This is another picture of a heterophyes worm. This is the oral sucker. This is the ventral sucker. And this is the gonotile or genital sucker. This is another picture of the heterophyes. And I want you to take a look at the red arrow. The red arrow is pointing to the uterus. And uterus in trematodes is known to house or contain eggs. And please do appreciate that the uterus is linked to the gonotile, because the main purpose of the gonotile or the genital sucker is to release the eggs of the parasite. This is the picture of the male and female Schistosoma adults. The bigger worm is the male worm. Can you also see oral suckers and ventral suckers in these worms? Yes. Take a look at the structure labeled as gynecophoric or gynecophoral canal in males. They will use this particular portion of their body to house or hold the female worm. We have already discussed the structures that function for attachment. It's now time to discuss the structures that belong to their digestive system. The red arrow is pointing to the oral sucker, and it is used by the worm to acquire the food from the external environment. Then the food will be swallowed and it will lodge on the structure pointed by the blue arrow, and that is the pharynx. The pharynx is continuous to the inverted Y-shaped structure, and that is the intestine or cecum. Take note, the intestines of trematodes would end blindly, meaning they will just terminate within the body of the worms. In humans, our intestines are linked or continuous with our anal canal. These worms do not have anus. Therefore, their intestines will just terminate or end blindly. For you to understand that, take a look at this worm. Look for its oral sucker. Immediately after the oral sucker, you have there the pharynx. Connected to the pharynx is the inverted Y-shaped intestine. And take a look at the length, the full length of the intestine. You can clearly see that the intestine will just terminate or end blindly because it's not connected to an opening. Just imagine this. These worms do not have ani or anus. So, they will also release the waste material through their oral sucker. So it's like eating burger, digesting the burger and converting it into poop and releasing the poop through your mouth. So, at this point, I want you to memorize the structures that belong to the digestive system. The oral sucker, the pharynx, the inverted Y-shaped intestines that will terminate blindly. Let's try to identify those parts in this particular trematode. Take a look at the body first. It's leaf-like in shape. It is dorsoventrally flattened, but it's not segmented. This is most likely the ventral sucker. This is the oral sucker. This is the pharynx. And attached to the pharynx, on both sides of the body of the worm, you have there the intestine. Take note, the intestine of this worm is unbranched. Let's check this fasciola worm. This is its oral sucker. This is its pharynx. And this is its intestine. What do you notice? The intestine of fasciola is branched. So meaning, all trematodes have simple or unbranched intestines except for fasciola. And you can use that particular distinguishing characteristic of fasciola to identify it under the microscope. So let's review. All have two types of suckers, the oral and ventral, except for heterophyes because heterophyes has an extra type of sucker, and that's the genital sucker. All have simple or unbranched intestine except for fasciola. These are additional pictures of fasciola worms showing us the branched intestines. In the case of Schistosoma species, their intestine will also bifurcate. But unlike in the case of most trematodes, the bifurcated intestine will later fuse at the distal end of the worm. That's why you have there the portion of the intestine that is labeled as reunited intestine or reunited ceca. Again, in the typical trematodes, they have inverted Y-shaped intestines. In Schistosoma, their intestines will bifurcate but will later fuse or reunite at the distal portion of the worm. At this point, let's talk about the excretory system of the worms. And the excretory system is also known as protonephridium. This is equivalent to the urinary system of humans. In our urinary system, what forms the urine to eliminate waste products is the kidney. The kidney will drain the urine to the ureter, and the ureters will drain towards the urinary bladder. In the case of trematodes, what will act as kidney is the flame cell. So you look for the flame cell in the picture. The flame cell will eliminate the waste products, and the waste products will be drained into the collecting ducts. We have there the anterior and posterior collecting ducts, and they are the ones equivalent to our ureters. And the anterior and posterior collecting ducts will drain towards the excretory bladder, and that is the urinary bladder in humans. These are the flame cells of the fluke or trematodes, and they will drain the waste products towards the collecting ducts, and these collecting ducts will later drain into the excretory bladder. Again, you have the flame cells. Flame cells will drain into the collecting ducts, and the collecting ducts will later drain into the excretory bladder. And the excretory bladder will eliminate the waste products through the excretory pore. And that is found at the distal end of the worm. The green-colored structures are representing the structures and organs that belong to the excretory system of trematodes. So it's very obvious in the photo that the flame cells and the collecting ducts are found at the lateral edges or sides of the worms. And the collecting ducts will all merge towards the center at the distal part of the worm so that they can form the excretory bladder. And the excretory bladder will release the waste materials through the excretory pore, which is found at the distal end of the worm. So if this is a trematode, this is most likely the excretory pore. In this particular worm, this is one of the collecting ducts. This is the excretory bladder. And this is the excretory pore. Trematodes do not have brains and spinal cords like humans. But these worms are provided with nerves so that they can also respond to their external environment. I want you to take a look at the oral sucker. Immediately after the oral sucker, you have there the pharynx. Do you know that there is that group of nerves around the pharynx of these worms? And they are labeled there as anterior ganglion. Or in the other picture, they are labeled as anterior nerves. So these are the equivalent structures to the brains of humans. Take a look at the second picture. From this anterior ganglion or anterior nerves, you will have their branches. And we will name the branches as ventral nerve cord, dorsal nerve cord, and lateral nerve cord. So these are basically equivalent to the nerves, peripheral nerves that we have in our body. Now, we will discuss the reproductive system of trematodes. Please remember that all trematodes are hermaphrodites except for schistosoma. So expect that in a typical parasitic trematode, you will have both the testis and the ovary. If you will be asked to identify and describe the testis of a typical parasitic trematode, always look for the testis at the posterior end of the worm. And it's easy to identify the testis because they would always come as pair. Now, testis of trematode worms can vary in shapes. Some of them might be round in shape or globular in shape. Some of them might be branch appearing or dendritic. And some of them could be lobulated or lobe. So, I will be showing you sample pictures of worms. We need to identify where are the testes. And after identifying, we need to describe whether the testes are globular or round, branch or dendritic, or lobulated. So let's try to identify where are the testes in this worm, and we need to describe them as well. So I'll give you 3 seconds to look for the testes. Remember, they are always found at the posterior ends of the worm and they always come in pair. These are the testes of this worm. Can you please describe them? Yes, globular or round. How about this one? This is one of the testes and this is its pair. Can you please describe them for us? Yes, the testes of this worm are branch or dendritic. How about for this worm? This is one of its testes and this is its pair. And then, take a look at them. They look like the nuclei of neutrophils. And the nuclei of neutrophils are described to be lobed or lobulated. This worm also has lobulated or lobed testes. In terms of their female reproductive parts, these worms have a single ovary. And the ovary is most of the time found anterior to the testis. And just like the testis, the ovary can be globular or round, can be branched or lobulated. This is Fasciolopsis busci. Take a look at its testes. They are highly branched. And as mentioned a while ago, the ovary is usually located anterior to the paired testes. So this is the ovary of Fasciolopsis busci, and it is branched similar to the testes. This is the ovary of this worm. Can you please describe it for us? Yes, it is globular or round. Take a look at the structure that is labeled as ovary in this worm. Can you please describe it? Yes, it is lobulated. Now take note, there is that structure that is found between the ovary and the testis, and that is the seminal receptacle. That is where the testis would drain the sperm cells of the worm. You might appreciate the seminal receptacle in some worms, but sometimes there's a little difficulty in identifying it in other worms. This is one of the testes of the worm, and it is lobulated. This is its single round ovary. And there is that structure where the seminal receptacle will drain the sperm cell in two, and where the egg cell is drained by the ovary in two. And that structure is called the ootype. This ootype is surrounded by glands, and we call the glands as Melis glands, and they are the ones labeled in the picture. Another structure will also drain into the ootype. So we now have the seminal receptacle, the ovary, the Melis glands. There's a fourth one, and that fourth structure is called the vitellaria. Now, what's the purpose of these four structures draining into the ootype? The seminal receptacle will drain the sperm cell, the ovary will drain the egg cell, the sperm will fertilize the egg, and you will now have there the production of the embryo. And this embryo has to be protected. That's why there's a need to produce the shell. And what will now produce the shell are the Melis glands and the vitellaria. And that's basically the reason why they also drain their secretions towards the ootype. Now, if you have the embryo and the shell, the egg now is formed, and the egg is temporarily stored in the uterus. And this is the uterus. And appreciate that the uterus is continuous with the ootype. This is the testis. This is the ovary. And then the seminal receptacle and the ovary will drain into the ootype. The ootype is surrounded by those Melis glands. Then this is your vitellaria, and this vitellaria will also drain into the ootype, so that the Melis glands and the vitellaria will form part of the shell. So I want you to recall, where can you find the vitellaria and the uterus? So most likely this is the uterus of the worm, and this is its vitellaria. So I hope at this point you can already identify the testis, the ovary, the vitellaria, and the uterus. In the case of Schistosoma, they are dioecious, meaning they have separate sexes. In the case of males, I want you to listen very carefully. Their testes are not found at the posterior end, because what they have on their middle to posterior portions is the gynecophoral canal. So their testes are found behind their ventral sucker. So take a look at the picture of the male worm. It has the oral sucker and the ventral sucker. Behind the ventral sucker, you have their rows of testes. And that's for the male schistosoma adult. In the case of females, the smaller worm in the picture, their ovary is found in the midline. Why? Because they don't have the gynecophoral canal. So that's why they can utilize their posterior ends, the middle to posterior ends, to house their other organs. These are two male schistosoma adult worms. The structures labeled with OS are the oral suckers, VS are the ventral suckers, TE are the testes. As you can see, they are found on the anterior end of the worm and immediately behind the ventral suckers. GYN is referring to the gynecophoral canal. And that's basically the reason why the testes have to be located on the anterior end of the worm, because what is found on the middle to the posterior portion of the worm is the gynecophoral canal. The bigger worm is the male schistosoma. The smaller and longer one is the female. Focus on the male worm. Locate its ventral sucker. Behind the ventral sucker are the rows of testes. These are the different stages that the trematode worms would have to undergo through in their life cycle. Ova, and the ova will hatch to release the meracidia. The meracidia will infect the snails. As soon as the meracidia will infect the snails, they will lose their cilia and they will become the sporocysts. Majority of them will give rise to rhidia inside the snails, and the rhidia will produce the tailed stage cercaria. The cercaria will emerge out of the snail and insist itself in the second intermediate host to become metacercaria. All of these stages are present in a typical parasitic trematode, while two of them are not seen in schistosoma. You already know one of them, and that's the metacercaria. So we will know later on what other stage is also absent in schistosoma. Ovum. The ovum will release the ciliated stage, which is the meracidium. The meracidium will penetrate the body of the snail, and once inside the snail, it will lose its cilia and it will become the sporocyst. Sporocyst will produce the rhidia, and the rhidia will produce the tailed stage, which are the cercariae. What will emerge out from the body of the snail is the tailed stage cercaria, and the cercaria will insist itself in the bodies of the second intermediate host, and it will become the metacercaria. And once ingested by humans, the metacercaria will mature to form the adults, and these adults will produce the ova, and the life cycle will again continue. This is a picture of an ovum that has already opened up its operculum because it's about to release the ciliated stage, which is the meracidium. This one is showing us the emergence of the ciliated stage meracidium from the egg through the operculum. This is another picture of the ciliated stage, which we refer to as the meracidium. This is the meracidium, and this is the one that will infect the snail host, except for Clunorchis and Opistorchis, because their infective stages to their snail host are their embryonated ova. I want you to listen very carefully. You have here the meracidia with their cilia, and they are capable of penetrating the body of the snail host. As soon as the meracidium will invade the body of the host, it will start losing its cilia. And once inside the body of the snail, it will now be called as a sporocyst. This is another picture of meracidia that are invading the body of the snail. As I have told you a while ago, as the merasidium will invade the body of the snail, it will start losing its cilia. And once inside the body of the snail, it will now be enclosed in a sac-like body, and you will refer to it as sporocyst. Let's review. Blue arrow is ovum. Yellow arrow is merasidium. And once the merasidium is already inside the body of the snail, it will lose its cilia and it will be enclosed in a sac-like body. You will call it as sporocyst. The sporocyst will eventually give rise to the rhidia. Now, what is now the difference between a sporocyst and the rhidia? The rhidia has almost the same morphology as that of the adult. Why? Because it has an oral sucker and a short digestive tract. So meaning, this rhidia can feed. And what do you think they will eat? I will discuss that later on. So, orange is sporocyst because you don't have there the oral sucker, while green arrow is a rhidia because it has an oral sucker and it is designed to feed. And another thing is, inside the rhidia, you have their developing tailed stages we call as the cercariae. Black arrow is pointing to the tailed stage, which is the cercaria, while white arrow is pointing to the encysted metacercaria. Beside the metacercaria, you have there the adult worm. This is a sporocyst. As you can see, it is enclosed in a sac-like structure and you do not see any oral sucker. This is the rhidia stage of trematodes. What do you notice? It has a mouth, that's the oral sucker. It has a pharynx, and it has a short intestine. Because the rhidia stage of trematodes is designed to eat. I want you to imagine, this rhidia is found inside the body of the snail. So this rhidia, once it is formed, will actually feed on developing sporocysts of other trematodes. And that is to get rid of the competition and to provide nutrients to the developing cercaria inside it. So once the trematode can form a rhidia stage, it would only mean that that trematode can eliminate other species of trematodes that are infecting the same snail. This is a picture of a rhidia. One is oral sucker, while 2 is the pharynx, and number 3 is the short intestine, while number 4 is pointing to developing cercaria. The sporocyst can produce cercaria, but it can also give rise to rhidia. I know it's confusing, but for now, it's enough that you know that the sporocyst can produce the rhidia. And as for the sporocyst producing the cercaria, I will explain that later on. Okay, for now, the sporocyst will rupture, releasing the rhidia. And the rhidia is identified by the presence of the oral sucker. The rhidia is mobile, and it can prey on developing sporocysts or the rhidia of other trematode species. Sporocysts can rupture and release the feeding stage, which is the rhidia. But some sporocysts can directly produce the cercaria, meaning they will not produce the rhidia stage or the feeding stage. And that is true to Schistosoma species. So meaning, in the life cycle of Schistosoma, they do not have both the rhidia and the metacercaria stage. So what will happen in the life cycle of Schistosoma is that after the merasidium will infect the snail, the merasidium will become the mother sporocyst. This mother sporocyst might rupture, releasing now daughter sporocyst, and this daughter sporocyst will now produce the cercaria. So Schistosoma is a little friendly to the other trematodes because it will not produce the feeding stage which might feed on their developing sporocyst or rhidia. So therefore, additional exemption to the rule or general characteristics of trematodes. All parasitic trematodes have the rhidia stage except for Schistosoma. At this point, it's very important to classify Cercaria based on their morphology. If they have long tails, you will refer to them as Lophocercus or Gymnocephalus. So the two Cercariae on the right side of the screen are Lophocercus or Gymnocephalus. The Cercaria in the middle has a short tail and you will refer to it as Microcercus. Microcercus, short tail. While the Cercaria on the left side of the screen has a forked tail. So therefore, you will refer to it as Furcocercus. There are Cercaria that are provided with long tails, but their tails are provided with fins. So we do not just simply refer to them as Lophocercus. We will call them as Plurolophocercus, meaning they have long tails and they have fins on their tails. This is the life cycle of Fasciola hepatica. Please take a look at its Cercaria. Classify it based on its morphology. Yes, you're correct. It is a Lophocercus or a Gymnocephalus Cercaria. These are pictures of Gymnocephalus or Lophocercus Cercaria. As you can see, they have long tails. This is the life cycle of the Schistosoma species. Please take a look at the cercarial stage. Can you please identify it or classify it based on its morphology? Yes, you're correct. It's Furcocercus. Please do not forget from now on that the cercarial stage of Schistosoma species is Furcocercus type of Cercaria. These are sample pictures of the Furcocercus Cercaria. This is the life cycle of the only lung fluke, meaning we can find the adult in the lung tissue. This is Paragonimus westermanni. Take a look at its Cercaria on the number 5 part of the life cycle. Can you classify it? Yes, it has a short tail. Therefore, this is a Microcercus type of Cercaria. This is a good picture of a Microcercus Cercaria. This is the life cycle of the Opistorchis species. Take a look at the Cercaria and please classify it based on its morphology. It has a long tail and the long tail is provided with fins. Therefore, this is an example of Plurolophocercus Cercaria. Take a look at the tail of this Cercaria. Then, you can see there the presence of fins. So therefore, this Cercaria is a Plurolophocercus type. The Cercaria will insist itself on the bodies of the second intermediate host and will become the Metacercaria, the infective stage 2 humans. Do you know that the structures in the Metacercaria are similar to the Cercaria? But there's one structure that is missing, and that is the tail. The tail is lost during the encystation process. This Metacercaria can be found in freshwater fishes, can be found on snails, can be found on aquatic plants. So later on, I will be teaching you that the second intermediate host of the typical parasitic trematodes will differ from one trematode to another, because the only thing that is common to them is the first intermediate host, and that is the snail. So in this picture, you have here the involvement of a freshwater fish. Eating this one raw will predispose us to develop the infection caused by the trematode or parasite. Some Metacercaria can be found encysted on the freshwater plants or aquatic plants. And shown on the right side of the screen is a picture of one metacercaria, encysted metacercaria. And that ends our lecture on the introduction to trematodes. Thank you for listening, future doctors.